The next structure is the cytoskeleton, and the cytoskeleton is the same amongst prokaryotic and eukaryotic cells, so we'll just kind of talk about it all at one time in the eukaryotic lesson. The cytoskeleton is a series of different tubules and filaments that run throughout the cytoplasm of a cell. It is composed of three different filaments: actin filaments, microtubules, and then intermediate filaments. And each of these have their own function. As far as actin filaments are typically found right underneath the cell membrane on the interior of the cell. They surround the entire surface of the underneath the cell membrane within the cell, and they are responsible for allowing shape changes and movement. So we can see in this image, if the cell membrane's right here in this kind of tan color, right underneath in the red, we see a line of these actin filaments. We see three actin filaments right there. And what those actin filaments can do is kind of shrink and then expand, and that allows the cell to change its shape, maybe from going more of like a circular shape to like an elongated oval, almost like maybe like rod type appearance. What it can also do is allow for a kind of crawling type movement. As the actin shrinks and then expands, that can allow the eukaryotic cells to kind of crawl along a surface. So various functions for actin filaments. Microtubules are long hollow tubes that run throughout the cytoplasm and are responsible for moving RNA and vesicles, like the transitional vesicles and the condensing vesicles, from one place to another. So we can see the microtubules actually really clearly here. So this is a whole cell. This blue is the nucleus within the cell, and all these green glowing filaments are the microtubules. But they run throughout the entire surface of the cell, and their job is to move RNA from one place to another and move vesicles from one place to another. So if we need to transport a vesicle, a transitional vesicle, from the endoplasmic reticulum to the Golgi apparatus, it just gets put on the appropriate microtubule, and it essentially rides that microtubule like a train on train tracks till it gets to the location. Or if we need to send RNA from the nucleus, where it's just been made during transcription, to a ribosome in the cytoplasm, again, same thing. It just goes on the appropriate microtubule and just rides that microtubule until it gets to its location, which in that case would be a ribosome. So the microtubules are responsible for transport of RNA and vesicles throughout the cell. And the last is the intermediate filaments. These are rope-like structures that are responsible for anchoring all the organelles to the cell membrane, so they're not just constantly being stirred up in the cytoplasm solution. So if we see right here, we have a mitochondria right here, and these little blue intermediate filaments are extending down from the mitochondria, and they're anchoring that mitochondria to the cell membrane, so it's always designated in that place. So now we're going to focus on specific types of eukaryotic cells. So most of what you know about eukaryotic cells probably extends to things like humans, plants, and other animals. But being that this is microbiology, we're going to focus on microorganisms, which are also eukaryotic cells. So that's going to be fungi and protozoa. So the study of fungi is a field in biology known as mycology. And mycology can be broken down into three different classes of fungi, which would be the yeast, the molds, and then the fleshy fungi, which are the mushrooms. Fungi are unique because they're able to survive in harsh environments. By harsh environments, this would typically refer to organisms that can survive in low or acidic pHs. That's abnormal. Most microorganisms prefer a neutral pH, but fungi are an exception to the rule, and they survive quite well in acidic pHs. And then also environments that have a high salt concentration. Most microorganisms, if you expose them to salt, it dehydrates them, they shrivel and die. That's why salt is used as a preservative. But fungi are unique. They actually prefer a high salt concentration in order to thrive. And that's why a lot of the various skin infections that we associate with fungi, like athlete's foot, toenail fungus, jock itch, those types of things, are all prone to occurring in areas of our body that have high sweat concentrations, because the fungi enjoy the high salt concentration, they thrive. And then our skin collectively has a low or acidic pH, so they do quite well in the skin and cause a variety of different infections on the skin. There are thousands of different species of fungi. Only relatively few of them, 270, are able to cause disease in humans, and we'll focus on several of these throughout the course of the semester once we get to the infectious disease chapter. There are many fungi that are non-pathogenic and many that are even considered normal biota to us and are beneficial to us. Fungi are what we call chemoheterotrophic, which means they are other feeders. They feed off of other living things in order to obtain carbon as their essential nutrient. So fungi feed off of humans, plants, animals, other bacteria, protozoa, fungi, just other living things in general, in order to obtain carbon as to generate energy. Technically, we as humans are considered chemoheterotrophic. We feed off of other living things like plants and animals in order to obtain our carbon source. So the three different classes of fungi, the yeast, the molds, and the mushrooms, can be divided into two subgroups based on size. So mushrooms are larger. You can see them with the naked eye. We would put them in the macroscopic fungi class or subgroup. Most molds and yeast are considered microscopic. The individual cellular structures we can only see with the assistance of a microscope. Because they're smaller, they are put into the microscopic fungi subgroup. We'll talk about yeast first. With yeast, they typically do have an oval shape, which we can see in both of these images here, so they're known for having an oval appearance. When a yeast reproduces, it reproduces through a mechanism known as budding, which can be seen very well in this top picture. So if a yeast cell wants to replicate itself, it basically doubles its internal contents, and then it pushes an entire new yeast cell out of its cell membrane. So we can see budding in various stages. So this yeast is kind of newly budding, so we just start to see the formation of a bud. And then this one over here is definitely more advanced. Eventually what will happen is the cell membranes of the two cells will kind of pinch off inward, and two yeast cells will be generated. So one will become two, and then those two will eventually divide and produce four. So that process is known as budding. And sometimes when they go through budding, they do completely separate from each other, but sometimes they stay stuck together after a budding cycle and form a chain. So when a chain of yeast forms after budding because they didn't disconnect, that is called pseudohyphae. It'd be very similar to what we see with bacteria when we talked about streptococcus. It would look very similar, but we're talking about different microorganisms now, so there's different terminology. When yeast stay stuck together and form a chain after a budding cycle, that's called a pseudohyphae. A thing that yeast are kind of famous for is their ability to produce alcohol. That's why they're used in the food and beverage industry. Yeast are what we call facultative anaerobes, which basically means they are able to survive in the presence or absence of oxygen. Later on the semester, we'll learn about different oxygen terminology. For example, if an organism requires oxygen, we say it's aerobic. If it doesn't require oxygen. We say it's anaerobic. But there are some organisms that can survive in both types of environments, so they can kind of go either way. And that's the facultative anaerobes, of which yeast fall into that category. So when yeast are exposed to oxygen, they'll generate energy, go through oxidative phosphorylation. But when you take oxygen away from them, in order to generate energy, they have to go through fermentation. So when you take oxygen away from yeast and they go through fermentation, a byproduct of them producing energy is producing alcohol. Well, we have obviously learned to benefit from that and have extracted alcohol for the beer and wine industries. So yes, yeast are kind of famous for being able to go through that fermentation cycle and ultimately leading to the production of alcohol. And again, that happens when you take oxygen away from them. Another type of fungi are the molds. Now, molds are different as far as shape than yeast. So if we look at yeast, they are more oval in shape. When we look at molds, as far as their shape, they're more long, thin filaments. If you've ever seen mold, it always has this kind of fuzzy, like hairy-like texture and appearance. And that comes from the idea that they are these long, thin filaments. So mold is made up of long filaments. These long filaments that we're seeing in both of these pictures are called hyphae. So a mold is just made up of long filaments called hyphae. And these are both pictures of different types of mold hyphae. Now, some molds within their hyphae, they can have these little cross walls or these little sections where the hyphae are divided into different compartments. These little lines or these little sections within the hyphae are called septa. So it really are just these dividers that divide the hyphae into distinct units. So if a mold has a hyphae that does not have any septa, so if it's just hollow all the way through, just one long continuous hyphae, we actually say that that is coencytic hyphae. It does not have any septa present, and that's what we're seeing here. However, if the mold does have hyphae that have the cross walls or septa present, as we see here, that is known as septate hyphae. So basically, molds are composed of long filaments called hyphae, and there's two options as to what they can have. Septate hyphae, which are going to have the septa present as the cross walls, and then coencytic hyphae, which are just going to be long, hollow filaments. No septa present. The last type of fungi are the fleshy fungi or the mushrooms, which are macroscopic. You can visibly see them. As far as mushrooms, what they're made up of is called mycelium. So a mushroom is composed of a mycelium. That's what makes up the mushroom. A mycelium is really just a mass of hyphae all intertwined, and that's where we get mycelium. And there's two different types of mycelium. There's vegetative mycelium that's found underground and acts as the root system to absorb the nutrients for the mushroom. And then up here above ground, we have the reproductive mycelium, which stores the spores, which are the reproductive units of the fungi. So within here, there are spores being stored. If these spores would be released and released into the environment, new mushroom growth would propagate in wherever the spores were located. So collectively, mushrooms are composed of mycelium, which just is a mass of hyphae. There's two different types of mycelium, the vegetative mycelium, which absorbs nutrients, and then the reproductive mycelium, which stores the reproductive spores.